Okay. So I cannot on my this video because the alarm starts to it triggers the alarm in the house. I don't know why. It's a connection, something wrong with the connection. So uh, you have seen me just now, right? And then I will just off the video, yeah, and you just hear my voice lah. Okay. So anything you can ask or you can just write in the chat box. All right. So we'll continue with the lecture. Okay. So let's see. I've shared. Uh, okay. Right. All this I've covered. Yeah. Okay. Right. Action of drugs. Yeah. So action of drugs. Yeah. Basically. Yeah. The action is quantitative. Yeah. But not qualitative. Yeah? They either increase or decrease the rate of function. Okay? For example, I mentioned yesterday, yeah, it could increase the heartbeat or decrease the heartbeat, increase respiration, depress respiration. Okay, so for example, like atropine, yeah, it is being used, yeah, to reduce the contraction of a smooth muscle of small intestine, yeah, so therefore it reduces bowel movement. So atropine, yeah, sometimes being given, yeah, to control diarrhea in human. But in our hospital, we don't use atropine. There are other drugs, yeah, used for diarrhea, yeah, in animals. And another example would be like aspirin. Yeah, aspirin decrease the pain sensation, right? Okay, site of action. Site of action, yeah. It can the interaction, yeah, the drug, yeah, when it interacts in the body. It could be at the extracellular, at the cell membrane, or intracellular. Okay, so example of extracellular is a alkaline salt, yeah, used to neutralize hydrochloric acid in stomach. So, like patient, yeah, having like excessive gastric juice secretion. Yeah, so alkaline salt is being used to neutralize the HCl acid in the stomach, and this process is referred as neutralization, which happens extracellular inside the cell. Cell membrane, yeah, uh, many drugs bind to receptors, yeah, to produce effect. For example, acetylcholine, yeah, binds to the receptor at the cell membrane, yeah, and produce the effect. So the effect is contraction of muscle. Okay, so a lot of drugs, yeah, they bind to the receptors at the cell membranes. Okay, intracellular. Okay, here, this is for drug for organisms, yeah, pathogens, infectious agents, yeah, such as the intracellular pathogens. For example, salmonella, yeah, it is an intracellular pathogen. So therefore, the drug need to enter the cell to produce its effect. So for example, fluoroquinolone, yeah, it's antibiotics, yeah, used for intracellular pathogens. Okay, drug transportation across membranes. Yeah. So once the drug is being taken orally, yeah, it need to cross membranes, yeah, in order to be absorbed, yeah, and then being distributed, yeah, to the target organs. So passive diffusion is the most important process, yeah, Here, drug molecules from the high concentration, it's the movement of the drug molecules from high concentration to low concentration. So no energy or carrier molecule involved. So rate of diffusion, is determined by the lipid solubility of the drug. Okay. Then the lipid solubility, further depends on the rate of ionization of the drug. Okay, I'll explain this. So the non-ionized form, okay, so drug, can form, it can be in the ionized form or non-ionized form. Okay, the non-ionized form will get across the membrane, to be absorbed and to produce effect. Okay, so let's look at this. So this I think this is a revision for you. You have learned this in your physiology, about passive diffusion. So drug diffuses, across the membrane in attempt to equalize, on both sides, of the membrane, from high to low concentration. From high to low concentration. So uh, to to be at equilibrium. Okay. So these are the definitions you need to know. You must know. I assume that you have known, but again, I'm just going to refresh you. With some of the common definitions. Okay, pH. pH. Is in reference to the number of free hydrogen ions. So that is the pH. When you say pH 1, so it's referring to the hydrogen ions. Okay, pKa. Okay, pKa refers to pH at which 50% of the drug is ionized and 50% is in the non-ionized form. So the ratio has to be 1 to 1. Okay. So please bear in mind that the pH of the stomach is acidic. And the pH of duodenum, the small intestine, it can range from 6 to 8. It's going towards more alkaline, pH of the duodenum, whereas pH of the stomach is acidic. Okay, so this one, just show you. Okay, actually, these notes are to explain this, what do you call, this this diagram. Okay, so acidic drug, So our concern here, drugs, they can be either a weak, usually they are weak acid or a weak alkaline. It's never a acid drug. It cannot be acid lah. Or else if you take an acid, it's going to destroy, your stomach and the other organs, esophagus and so on. So it is either a weak acid or a weak alkaline. Okay. So okay. So here, acidic drug. So there are two drugs, A and B. A and B. So the A is aspirin and the B is a sulfur dimethoxy. So sulfur dimethoxy is a sulfur drug, commonly used in chickens for coccidiosis. Okay. Uh, do you know what is coccidia? It's a parasite. Have you come across of coccidiosis? Anybody? Okay, never mind. So later on, you will know.
Yes.
You have, okay. So, but never mind. I will just, I won't go into detail. Later on, when I discuss about parasitic drugs, bacterial drugs, then I go into detail. This is just an example. So here we have two drugs, aspirin and sulfur dimethoxy, A and B. So you look at the A, Okay. So the cursor is working. You can see the movement of the cursor,
Yes.
Yes,
All
yes.
right, all right. So okay, so here, so acidic drugs, it's an acid drug, So aspirin, sulfur, both are acidic drugs, weak acid. So acidic drugs, they exist predominantly in the non-ionized form, This is at the non-ionized form, yeah, at any pH that is more acidic than the drug's pKa. So where is what is the pH, yeah, of the drug um, when what is the drug's pKa?
3.
3. Okay, good. Yeah. So pKa is 3. So you have understood my answer. So pKa 3. Why 3? So this is the time where the ionized form of drug is at 50%. Yeah, both are at 50%. That same ratio 1 to 1. Okay. So the ionized form is at 50% and the non-ionized form is at 50%. So the ratio is 1 to 1. So that is the drug's pKa for aspirin is 3. Okay. So it says that, yeah, for acidic drugs, yeah, so anything, yeah, below the drug's pKa, yeah, the non-ionized form will predominate. So if the pKa is 3, yeah, so anything below the pKa, that means pH, yeah, pH that is more acidic, yeah, more acidic than the pKa, which is like 2 or 1, yeah, they will be in the predominantly in the non-ionized form. Okay, so the ratio, yeah, the ratio changes predictable for each increment of the pH. Okay, as you can see here, yeah, after it reaches the pKa at pH 3, yeah, um, the the non the ionized form start to change. Okay, the percentage, yeah. So yeah, here the ratio changes predictable for each increment, which is like it will go down to, yeah, it will start to increase. The ionized form will start to increase at a factor of 10. Okay, 10, 100,000, 10,000, and so on. So you can see the colored column here. So as the pH increases, the ionized form increases by the factor of 10. Okay. Whereas for this drug, yeah,
Doctor, I have
yes,
question. That mean if the pH is below than 3,
mm,
so the drug cannot dissolve. Is it or not? I don't
no,
know.
no. Who said cannot dissolve? Now we are talking how the drugs are transported across membranes. It depends on the rate of ionization. So therefore, it was mentioned that this drug, yeah, below pH 3 is uh, uh, readily able to penetrate the cell membranes.
Okay,
Did
okay,
you,
understand.
did you see my previous lecture? I said the lipid solubility, it depends on the rate of ionization of the drug. So lipid soluble drugs, they will go across the membranes easily. Why why lipid soluble drugs can cross the membranes easily? Anyone? Physiology? Oh, the cell membranes are made of,
Lipid soluble.
yeah, lipid
By
soluble.
lipid.
Bilipid, phospholipid bilayer.
For yes. So that is the reason why I'm talking about lipid solubility, because the membranes are made of phospholipids. So therefore, only like if it's lipid soluble, yeah, they'll be able to cross easily. Yeah. So only the non-ionized form is in the lipid soluble form. So therefore, here, yeah, drugs, yeah, at pH in acidic environment, yeah, the non-ionized form, yeah, because the non-ionized form is at pH 1. right? So if the pH, so the drug, an acidic drug, even in an acidic environment, the absorption it's good because it can cross the cell membrane. Yeah. Okay. Whereas this one, another example here. Okay. I just want to okay. So B. Okay. B is a sulfur drug. Yeah. Example of a sulfur drug. What is the pKa here?
6.
Yes, 6. Okay, you understood. So pKa is 6, right? So anything, so this is an acidic drug, anything, below 6, right? Look at below 6, yeah, you can see the non-ionized form, right? It's increasing, right? So at pH 1, so I would say at pH 1, this drug, sulfur drug at pH 1, the stomach, that means, it is readily penetrate the cell membranes. Okay, as the pH increases, at 7 and 8, okay, when it's more in the ionized form, less penetration. So both of these drugs, so the conclusion from here is acidic drug, in the acidic environment, it is in the non-ionized form, and penetration through the cell membranes is good. Okay, so actually this one explains the previous slide. Yeah, I can show you the previous slide. How to go to the how to go back. Sekejap, sekejap, Okay, this one. Yeah. So actually, what I've mentioned just now, is written in the notes form. So acidic drug exists predominantly in the non-ionized form at any pH that is more acidic, than the drug's pKa. So if the pKa is 3, less than 3, they're all in non-ionized form, and penetration it's good, right? Across cell membranes. So the ratio predicts the ratio changes predictable for each increment of the pH change, and then the ratio changes by the factor of 10, for the increment of the pH change. So sulfur, if you compare sulfur and aspirin just now, so sulfur pKa dia berapa tadi? 6, and then the aspirin is 3. So sulfur methoxin can more readily penetrate the stomach cell membranes as compared to aspirin, because sulfur dimethoxy molecules are in the non-ionized form. Therefore, the lipid solubility property is very high. Okay? They're highly lipid soluble drugs. Right? So this difference, can be attributed to the drug's pKa, right? So the difference between the pKa, determines the transportation of the drugs across the cell membranes. Okay, right. So I've mentioned this, So drugs in lipophilic, which is the non-ionized form, are more readily absorbed after oral administration. Please bear in mind, after oral. When you give a drug IV, there's no any barrier. It goes direct into the blood circulation. It need not be absorbed. No absorption phase after intravenous administration. But with drugs given orally, intramuscular, yes, there is a phase of absorption, then followed by distribution, right? Okay, So nature of the drug, whether it's acidic, or alkaline, so it changes. So what I mentioned just now was acidic drug, so whether it's acidic or alkaline, um, it gives different results, different outcome. So the nature of the drug, the pKa, the pKa of the drug, which the percentage, 50% of the ionized and non-ionized form, which is the ratio 1 to 1, and the pH of the environment. pH of the environment here refers to the stomach and the small intestine. Intestine is more alkaline, whereas in the stomach is more acidic in monogastric animals. Okay, so what I'm talking here is mainly on monogastric. Usually, you give it oral, uh, it's better. The absorption is better in monogastric animals as compared to ruminants. Because ruminants has 4 stomachs, and in the rumen you have a lot of microbes. They can degrade the drug, so it's not really advisable in ruminants to give orally. Usually it's given IM, intravenous, and so on. But this in monogastric, yes, okay, because of the good absorption of the drug. Right? Okay, so this is just to show you that, okay, so as I mentioned earlier, weak acid, drugs are either weak acid or weak base. So weak acid, form a HA, so this is the uncharged form, right? And it can be in the charged form, the unprotonated form, okay? And a weak base, can be in the uncharged form, and with proton, or in the charged form. So this equation, is reversible. That's why I put the arrow on 2 sides. They need to be at equilibrium. Okay. So this is the non-ionized form, right? The uncharged form, non-ionized, and this is the ionized form. Whereas for a weak base, okay, this is the non-ionized form. Okay, this is a base drug. Okay, non-ionized. This form is ionized form. Okay, so Henderson-Hasselbalch equation, is used to calculate the percentage of a drug that exists, the ionized form, or to determine. So when you calculate the ionized form, definitely you can get the non-ionized form, right? So the non-ionized form, you can determine the concentration of the drug across a biological membrane. Okay, again, this is again I'm repeating, for so that you understand better. So acidic drug, again, aspirin. This is the molecular structure. So this is the non-ionized form. So this is the ionized form. Okay. So with the non-ionized form, you have the OH group. The ionized form, it loses its hydrogen ion. Okay. So these are the forms. So this is the non-ionized form. This is ionized form, right? These are examples. Okay, I've mentioned about the weak acid just now. So when in non-ionized form, this is in non-ionized form, so the weak acids, they donate, H plus, and they become ionized. Okay, so this is not good. Okay, right here, because as you know, acidic drugs are hydrogen ion donors, whereas base drugs are hydrogen ion acceptors. Okay, so here they be always like wanting to donate hydrogen ion, and become ionized, But in the acidic environment, yeah, where free hydrogen ion exists, so acidic environment, yeah, there are a lot of, because the pH is low, yeah, pH 1, 2, or 3, yeah, so there will be a lot of presence of hydrogen ion, yeah. So the ionized form, yeah, this form, the ionized form, yeah, they will encounter, yeah, the free hydrogen ions in the stomach, yeah, and combine with it and become non-ionized. Okay, so the non-ionized form is the one that can get across the membranes easily and easily absorbed into the blood circulation. Okay, right. So we go to the alkaline drug. Okay, alkaline drug example cimetidine. Okay, cimetidine used in veterinary medicine. Yeah, it's a drug for peptic ulcers. Ulcer in the stomach. Yeah. So this drug, okay, um, this is the molecular structure. Okay, so this is the ionized form, and this is the non-ionized form. Okay, right. So this is the base. Yeah, yeah. Okay. So it accepts the hydrogen ion, yeah, and become ionized. Okay. It accepts the hydrogen ion, H plus, and becomes ionized. Okay. So this is the base drug, yeah, right? When it accepts the hydrogen ion, becomes ionized. So a weak base, yeah, which is an alkaline drug, yeah. So in acidic environment, so it's given in the acidic environment in the stomach, when it enters the stomach, yeah, the non-ionized molecule of the alkaline drug, yeah, will combine with the H plus and becomes ionized. Okay. So this is the non-ionized, the unprotonated or uncharged form. Yeah, will combine, yeah, with the H plus becomes uh, becomes protonated or charged. Yeah. So the charged form is the ionized form. So the percentage of the ionized form increases. Okay. So therefore, the absorption of a base drug, yeah, in the acidic environment, A is it's lower as compared to an acidic drug. Okay. So this is an example, right? Just now I showed acid drug. Now alkaline. Alkaline, okay, I have diazepam. Yeah, diazepam is a drug used for convulsions, yeah, to control convulsions in dogs. Okay, and lidocaine. Lidocaine is a local anesthetic drug. Okay, both of these drugs are alkaline, and actually, I'm not going to explain this. Yeah, the concept, yeah, is very similar to acidic drug. Yeah, pH, the pKa, yeah, the pKa is 3, and then here the pKa is 8, yeah, and I think I will post this question, yeah, I'll post this question in forum, yeah, later on you can try to answer, yeah, uh, answer the question and write your answer in the forum, yeah, I'll create a forum so everybody can try, yeah, to answer question. Yeah. So the question is explain the ratios of the ionized to non-ionized molecules of diazepam and lidocaine at different pH values. Okay. So explain, yeah, as I explained just now, yeah, how is being forms ionized and non-ionized, yeah, both the drugs, and later on compare, yeah, compare the rate of penetration, yeah, through the cell membranes. Rate of penetration of diazepam and lidocaine. Okay, right. So this is another form, yeah, to explain the ionized and non-ionized form. Yeah, this is in the graph form. Yeah. Okay, it's a very similar concept. Yeah, same concept. So as for weak acids, yeah, okay, this is the pKa, yeah, okay, right. So when the pH is less, yeah, than the pKa, right? So which means this, okay, when the pK is less, yeah, this is the pKa less, okay. The protonated, the non-ionized form predominates. So this is a graph that shows the weak acid, right? This is a weak acid, and this is the weak alkaline, right? So when the pH, yeah, is less than the pKa, yeah, the non-ionized form predominates. Okay, as you can see here, the percentage of non-ionized, so that 100%. Right. So when the pH, yeah, is more than the pKa, so it's going to the right. Yeah, look at this graph. Yeah, going to the right. The ionized form predominates, right? And same thing, yeah, for the base, weak base. So when the pH, yeah, is less than the pKa, yeah, the ionized form, yeah, because it has got the charge, yeah, H plus, the hydrogen ion, yeah, it predominates. And when the pH, why is it very low here? Because this graph, yeah, the plot here is a non-ionized form, right? So this one has to be very low lah, okay? Because this is a non-ionized form. So it refers to this and this actually, HA and B. So the B is the non-ionized form, which will be at 100%, which predominates. Okay, if the pH is more, yeah, than the pKa, the non-ionized form, yeah, predominates. Okay, that is for a weak base. So that's a difference, weak base and a weak acid. Okay, so when you explain, yeah, you need to explain about, when I asked you to explain about the rate of ionization of drugs in general, so you have to explain in two aspects, weak acid and a weak base, right? Okay, right. So that was on passive diffusion. Please remember, yeah, most of the drugs, they go across through passive diffusion. So the other methods of drug transportation across membranes are, one here is facilitated diffusion. Yeah, we have just now passive diffusion. This is number two. Facilitated diffusion. This requires an attachment to a carrier molecule, so no energy is involved. Yeah. So, for example, like vitamin B12, yeah, it's transported by a glycoprotein. Usually, it's being attached, yeah, to a protein, yeah, and being transported, yeah, across the cell, across the membranes into the blood, okay, to the blood circulation. And active transportation, yeah, this one as the what we call the name, yeah, active, yeah, that means you require energy, yeah. You can imagine you want to do exercise, be very active, so you need energy, so it requires energy and a carrier molecule. So this one, it operates against a concentration gradient, which means it flows from the uh, uh, low concentration to high concentration. Yeah? It's like climbing a staircase, right? So example, yeah, it's an anti-cancer drug, yeah, 5-fluorouracil, yeah, it operates against a concentration gradient. Okay, this is just to show you, yeah, the active transportation, yeah, where the GIT lumen, yeah, the drug is being, yeah, it being transported, yeah, with a carrier molecule, yeah, and forms a complex and into the blood circulation. And yeah, the other methods would be these are the other minor methods, yeah, filtration, through pores. Yeah. So filtration is through the pores, yeah. Um, Usually large molecular weight substance, yeah. Hang on a minute, too fast. Okay. Okay, this is basically through the glomerulus. Okay, so which has got large pores. So they will allow large molecular weight substance, but not albumin. Okay, let's see. Pinocytosis. Okay, this one, not many drug molecules, yeah, undergo pinocytosis, very minimal. So drug molecules, they enter the cell and then they're being engulfed by the cell. So this is the active process, requires energy. So mainly like polypeptides, yeah, bacteriotoxins, antigens, yeah. These are the ones that go through pinocytosis. It's not carrier mediated. Right. Usually when the drug, yeah, unable to go through the membrane, yeah, or pass through the membrane, they will undergo pinocytosis. Okay, next, yeah, it's on types of membranes, yeah. Okay, this is you need to know, yeah. This is very basic pharmacology. So, blood-brain barrier. Have you heard of this blood-brain barrier structure in the brain?
Yes.
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 Okay, this is very important in pharmacology. These membranes, between the blood and brain, they have pores, yeah. They prevent many materials, yeah, often toxic materials from entering the brain. So it's well protected. The brain is well protected. Small lipid materials, yeah, or lipid soluble materials such as diethyl ether, halotane, yeah. These are the drugs that can easily enter the brain. Okay, why? They are highly lipid soluble. So as for like to enter the barrier, yeah, the drug, yeah, has to be highly lipid soluble. So only drugs such as I mentioned there, yeah, that is the halotene. All these are general anesthetic drugs. Okay, so they easily penetrate the barrier and produce the effect. So this is just to I think this is a revision. Yeah? Must have seen all these cells histology lectures. Yeah, to show you that the barriers actually, yeah, that are here, the tight junctions. Yeah, so extremely lipophilic drugs, yeah, will penetrate, yeah, the glia cells. This is the glia cell. You have glia cells, yeah, and also the capillary endothelial cells. So these are lipid soluble, yeah. They be able to penetrate the cells. So the hydrophilic drugs, they're unable to pass through the cell membranes. Okay, and then the renal tubules, as the kidney, there are a number of regions. Yeah, they're very important for drug elimination. So most of the drugs are eliminated via the kidney. So in tubules, drug absorption, reabsorption can happen. Okay. So membranes are relatively non-porous. Only lipid compounds or non-ionized drugs. These are also depending on the pH and the pKa are being reabsorbed at the renal tubules. So the concept is still the same. pH of the environment, the pH of the urine. Here the environment is urine. Okay, for elimination, it's excreted in the urine. pH of the urine, the pKa of the drug are important. Okay, blood capillaries and renal glomerular membranes. Okay, they are quite porous, allowing non-polar and polar molecules. Up to fairly large size, just below that. So if there is albumin can go through glomerulus, that means it's not good news. It's kidney failure. So come to indicate that you got problem with the kidney. So this is useful in the kidney since it allows excretion of many of the polar and waste compounds, substance. Okay, so summary, you can read that. So have you got a lecture after this? Can I go to the next slide? Can I share the next slide? Hang on a minute, Do you have a lecture after this?
Yes, doctor.
Yes. What time your lecture starts?
11.
11. Okay. You need a break? Wait, let me see. I'll take 5 minutes. Is it okay? You've got 10 minutes now. Can I give you like a 5 minutes break before you start the next lecture, or you need 10 minutes break? Try to share. Everyone is quiet. Where's is this the one? Let's see. This one. For the file, also taking 5 minutes. Okay, found it. Okay, I will just introduce a few words here. Okay, just very briefly. We'll just take 5 minutes. Okay, so basically, we'll talk about the pharmacokinetics. So this is going to be a general principle pharmacokinetics. Not going into detail. So I will talk about absorption, distribution, metabolism, and excretion. So the learning outcomes is just to expose you to the concept and principles of drug pharmacokinetics. So what is the, look, always I will start with the definition. What is drug absorption? So absorption is usually uptake or removal of the drug from the absorption site into the circulation. Okay. For example, from the muscle into, when you give an intramuscular injection, it hasn't gone into the circulation yet. It's still in the muscles. So it need to be absorbed. Into the, from the site of absorption to the circulation, to the blood. Drug distribution, it refers to the movement of a drug from the systemic circulation, to the site of action. And the site of action is usually the extravascular compartment. So it will move from the systemic circulation to the extravascular compartment. Okay, right. I will just show you this. So just to show you that, this one, so there are elementary routes, of drug administration, and also parenteral. Okay, so with elementary route, it's two routes, oral and rectal, and then with the parenteral route, the meaning of parenteral route, it circumvents the gastrointestinal tract. It doesn't go into the stomach. Okay, so these are the routes. IV. It goes right into the intravenous, intramuscular into the muscles, subcutaneous under the skin, intraperitoneal into the peritoneum. Intrathecal is under into the theca at the spinal cord. Intraarticular into the joint capsule. Okay, and we also have drugs that are given topical, on the skin, right? So on the skin, the degree of absorption is dependent on the lipid solubility. Volatile liquid, like halotane or gases anesthetic, are given through inhalation. Okay, for example, nitrous oxide. Okay, I'll stop here. Right. Any questions? Stop sharing.
Doctor,
Yes.
someone is asking question in the chat.
Okay, where's the chat now? Okay. For passive diffusion, passive diffusion, okay. If a drug is alkaline, it is become ionized in the stomach, then come back to is alkaline, it become ionized. Okay, so usually lah, like alkaline drugs, so we don't want it to be get destroyed in the stomach. So usually it has got a special coating, enteric coated tablets, it called as enteric coated, special coating where it will be released in the intestine for absorption. Okay, so these are called enteric coated tablets. Is that clear? Who is it? Chong? Okay. Right. Any questions before we meet on the Friday? Amy, any questions? So far so good. Can on your can unmute if you want to ask anything. Everyone okay? I didn't hear a response. I can't see you. You can't see me. All are fine. All are good today.
All good,
So far, so
doctor.
good, doctor.
So far so good, huh?
Okay.
Okay. Thank you very
Yes.
much.
Okay.
So I'll see you on Friday.
Okay, inshallah.
Okay.
Okay.
Thank you, doctor.
Okay,
Doctor.
thank you.
Okay, doctor. Thank you.
Okay. Bye. See you on Friday.
Thank you, doctor. See you.